All right, welcome back. Now we're moving into Unit 5, Molecular Biology. Like I said, this is some of my favorite stuff. I will restrict myself from going too in detail and nerding out on y'all. But this is what I did my PhD work in. I love talking about DNA and how it regulates the body, and it's really cool stuff. This is a really fun unit overall. So I'm going to break it up into several parts, little subparts to kind of help you get through it. What I'm going to do in this first lecture is talk about DNA structure, just to kind of refresh our brains on it, and talk about how we copy it. The next lecture, I will talk about transcription, how we go from DNA to RNA, and then a short lecture after that on translation, going from RNA to protein. And then we're going to have another short lecture after that talking about how we regulate genes and a little bit about mutations. And then after that, there'll be a lecture just solely on the biotech part. So let's get into some of the material. Now we're going to be talking about what's called the central dogma of molecular biology. But what I mean by that, we go from DNA to RNA to protein. So all the different proteins. So we talked about proteins, how they have so many jobs in your body. Enzymes is a good example. So maybe you need a certain enzyme in your stomach to break down something. So what we need to do is, we're running low on it. What we do is the cell senses that. It says, okay, we need to make more copies of this enzyme. So what we're going to do is we're going to go to the recipe, which is coded in your DNA. So that gene that codes for that protein is like the recipe. So what we're going to do when the whole central dogma is, we're going to go to that recipe. We're going to make a copy of it from DNA to RNA in a process called transcription. So pretty much like take this, make a copy, and then we're going to take a copy of RNA and use that to turn it into a protein, and that's the process of translation. Now it's going to be several steps. It's going to seem really confusing at first, but we break it down into several parts, and it's not going to be too confusing. This lecture is going to talk about this first part where we just copy DNA. So remember, if we're going to go mitosis or meiosis, we have to copy our DNA because we're going from 1 cell to 2 if it's mitosis, or 1 to 4 if it was meiosis. So I just want to give you a little bit of rehash on the anatomy of DNA versus RNA. So normally when we talk about DNA, it's going to be double-stranded because DNA does not like to be single-stranded. You're going to see when we get to talking about part of the replication, we have to make sure those nitrogenous bases don't close back up before we start copying them. Now, there are different nucleotides. Both of them are composed nucleotides, but there's going to be some difference when it comes to the bases. Now, there was a difference. We have a sugar called deoxyribose. That's the sugar that we use for DNA. For RNA, was a sugar called ribose. Now, the differences are in the bases. For DNA, we had the bases cytosine, guanine, adenine, and thymine. For RNA, we had cytosine, guanine, adenine, and the big difference right here is instead of thymine, we have uracil. Now, the ribose sugar in DNA, and then also the pairing of U, leads to RNA being stranded. It has a lot more flexibility with this backbone. So it's able to fold in on itself, form hairpins and loops that you'll see when we talk about translation especially. Now the base pairing rules, I'll type this in real quick. For DNA, C is always going to pair with G, and A will always pair with T. For RNA, it's going to be a little bit different. C is always going to pair with G, so that's equals G, sorry. And then A was going to pair with, sorry, keep pushing there, pair with U. So instead of the thymine that we have up here, we're going to replace it with uracil. Oh, click right there. So think about DNA replication. How hard can it be? You know, it's just making a copy. You're just going to put it in and make an exact replica. Now, it's Not that simple, it really is. There's a lot of enzymes. If you just look at this, you see all these different components. And I always think that like an orchestra, everyone has their part. You have to come in and play when you're supposed to. So the same thing's going to happen with DNA replication. We're going to have to open up the DNA strand, and then we're going to have to use each parental strand as a template and add the complementary bases to it. And then we're going to go from one strand to two strands. Seems quite simple, but it's actually a little bit more complicated than that. So here's a little bit more about the anatomy. So if you remember with DNA, the monomer is a nucleotide. Nucleotides consisting of a sugar attached to a phosphate group attached to nitrogenous base. And I mentioned the four bases for DNA. We have cytosine, thymine, guanine, and adenine. And the base pairing rules, C and G always paired together, A and T always paired together. Now, it's thanks to these constant base pairing rules that allows us for each parental strand to serve as a template. So that's why we're able to go from one strand to two quite easily. Now, DNA, you'll hear this term anti-parallel. It's really structurally stable, but it loves to be double-stranded. It does not like to sit in that single-stranded state very often. And it's thanks to those base pairing rules that, we will be able to use both strands as a template when we go through replication. Now, these bases are held together by hydrogen bonds. So we talked about the different type of bonds in Unit 1. Hydrogen bonds hold the nitrogenous bases together. The other type of bond that we will talk about is called a phosphodiester bond, and that's how we're going to connect the nucleotides. Now, thanks to how DNA is organized with how the nucleotides and where the phosphate is attached to a sugar, we're going to have to label the ends of it. So on one end, we're going to have what's called the 5' end, and it has to do with 1, 2, 3, 4, 5, how this phosphate is attached to the 5 carbon on the sugar. That's going to be our 5' end right here, and this is going to be the end of one strand. Now, when we want to grow a strand, we have to add it on to the 3' end. We cannot add on to the 5'. If I'm going to grow a strand of DNA, it has to be in this direction. So for this strand, if I was going to elongate, it can only go this way. If I was going to elongate this strand, since it would only go this way, which is going to make DNA replication seem a little bit odd because we're going to grow it in one direction and grow it in the opposite direction. It all has to do with the 5' and 3' ends. So if I'm going to grow my nucleotide strand, when I'm going to add on a new nucleotide, I can only add on to the 3' end. So if I wanted to extend this strand, I could only add over here. Now the opposite strand goes the opposite way. It has to do with how the base pairing rules line up and how the bases form up. So it's kind of flip-flop. So on this end, it's 5', but if you look at the prime end on this one, it's 3. So it's kind of like flip-flopped. This is where it terms anti-parallel. So it's going to come and haunt us a little bit later on. Okay. So here's another term that is going to DNA replication is semi-conservative. By that, I mean each parental strand serves as a template. The complementary base pairing rules make that possible. So here's an example right here. We have our parental strand labeled in this lilac purple color right here, this darker color. Now when we do DNA replication, pretty much what we do is we kind of break all those hydrogen bonds, boom, 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 and open it up and then just match the complementary bases to each one. And then we split it into 2. So if you look at this one, you see half of the backbone is that purple on each of these new strands, and half of it is orange. The orange is the new daughter strand. It's the same exact DNA sequence thanks to the base pairing rules. So they match up exactly. So this is semi-conserved because we conserved part of the original parent. So half of it's the original parent, half of it's a newly synthesized daughter strand. If you're interested in how this was figured out, this is a good video that talks about the experiments that were done to prove this theory. So it looks like a lot of words, but I use this as a good summary slide to show you all the different players and the big ones that I really want to focus on. So there's going to be a lot of proteins, but each of them has a specific job. This is a good review slide. And I think on one of the assignments, you actually have to go and identify what the job of the different enzymes do. So we're going to have a replication bubble. This is a bubble right here. And within that bubble, we're going to have two replication forks. And the reason we have that is we're going to start in one location. And we're actually going to go through replication in both directions. So we're going to be dividing or replicating this way and this way. So each fork will keep going and going and going until it reaches the end or encounters another replication fork. Now within here, there's going to be several enzymes that are going to have several jobs, or proteins. The first one's going to be DNA helicase. And the job of helicase is to unwind the double helix. So what it's going to do is be breaking down those hydrogen bonds between nitrogenous bases and making it from a double strand to a single strand. Now we have to get single strand so we can add in the complementary bases to make a copy. So a helicase is shown by this little triangle right here. Now, sometimes it's not shown right here. There's an enzyme called topoisomerase. It kind of runs ahead of helicase and eases some of the tension on the backbone. It makes it a little bit easier. It might make a nick here or there to help it break up a little bit. Now, DNA loves to be double-stranded. So what will happen is we will lay down what are called single-stranded binding proteins. They're not shown here. They'll be on the next couple of slides. And what they do is they prevent the bases from closing back on each other. And they're kind of like little temporary placeholders. Your first big guy that's going to come do a job is called RNA primase. What he's going to do is he's going to find the starting point, and he's going to lay down some RNA primers. And we have to do this because we just can't start laying down DNA nucleotides right away. We have to lay down some RNA first. It's just one of the quirks of DNA replication. So RNA primase is going to come through and lay down some primers. So it's shown on green on here. So you see several little spots of green. Those are going to be RNA primers laid down by RNA primase. Now the big workhorse is going to be this DNA polymerase 3, or sometimes it's called DNA alpha. And what this big old guy is going to do is he's going to be adding the complementary bases. So he's going to look for those RNA primers, hop on, and then he's just going to go along the strand and add in the complementary bases. That's his job. Afterwards, another guy comes in. So remember we added RNA bases right here. We need to get rid of them. So DNA polymerase 1, or sometimes called delta, is going to take away those RNA nucleotides and replace them with DNA. It's not shown here. And then finally, DNA ligase. So we're going to have these nicks in the backbone from the different sections, and I'll talk about what they are in a second. What he does is he fixes sections. So where does it all start? You know, you can just don't randomly just pick one place and go to the end. You would think for linear, we would just start here and work our way down. But if you're circular, what do? Well, we have what's called an ORI, or origin of replication. And it's normally going to be marked by a bunch of proteins, and it's a site where, okay, this is where you need to start replication. Basically, it's a start, and if you're a single, like a prokaryote, what you'll do is you'll open up, and then the replication fork will go both directions. And eventually, you'll get to where these two circles are completely copied, and they'll split apart, and we'll have two chromosomes. Now, there will only be one ORI for prokaryotes. For eukaryotes, we have these long linear chromosomes, and I always say work smarter, not harder. So what we actually have is multiple ORIs throughout our linear chromosomes. So you'll have multiple origins, and each origin will have a replication bubble. And the replication bubble will go both directions until it encounters another one. And once it encounters the next replication bubble, it'll stop, and then eventually we'll get to the two strands. So working smarter, instead of starting at the end and working our way through, we're going to start in multiple places, and then we'll work till you encounter another one. So there's going to be three big stages. We got to start it, we got to grow it, and we got to end it. That's pretty much how it's going to work. You'll see that those terms come up in transcription and translation. So with initiation, so here's a little bit more information about the different proteins and what they're doing that I introduced before, but focus mostly on this picture right here. So there's an ORI right here. This is only showing half of the replication fork. This is going on the opposite side, but just think of it being like a flip-flop. And remember that the strands are going to be different orientation over there. So what's going to happen is helicase, this purple guy right here, what he does is he opens up the DNA. He's breaking these hydrogen bonds between these bases, and helicase is just going to keep going. So the replication fork is going this way. So helicase is just going to keep going and going and going. And ahead of helicase, there will be this enzyme, we'll call that topoisomerase. I know it looks kind of weird right there, but consider this like a little ring that's running ahead. So helicase will keep opening and opening and opening. Remember, DNA loves to be double-stranded. So what's going to happen is these little purple balls right here, those are your single-stranded binding proteins. What they're going to do is bind to the DNA and prevent it from closing back up because it's kind of uneasy. It doesn't like to be single. It has to have someone bonded to it. So these will stay on there temporarily. Now that we have everything opened up, we are going to have to add on our primers. So we have to add like those little base coat bases to help us start. And this is done by RNA primase. So RNA primase finds a starting location, and then what it's going to do is going to lay down the RNA primers, which are shown in green right here. Now, if you remember, when we grow nucleotide strands, or when we're going to add new nucleotides, we only add it on to the 3' end. So since DNA is anti-parallel, we're growing from 5 to 3 direction. So if this is 3', our new strand that we make is going to have a 5' end, and then we can keep adding on bases this direction. Now remember, DNA is anti-parallel, so this strand, the bottom strand, has a 3'. So when we add on new nucleotides going 5 to 3, we're going to have to go this direction. So I'll talk about that in the next slide. But what's happening here is we're going to lay down primers, shown in green, and we're going to lay some down here. So this is like getting everything started and prepped. Now, thanks to the anti-parallel properties with DNA, we can only add on to the 3' end. So what happens is when we have a replication fork, we're going to have one strand that's going to be what we call our leading strand, and the other is the lagging strand. Now, the one that has continuous replication, so once it starts, it just keeps going, that's going to be our leading strand. It has the easier job because once it opens up, it just pretty much chases helicase and just adds on. Now, the opposite one, you have to do a little bit more work because it's like you're working backwards. So remember, DNA is opening up this direction, following the arrow. On the lagging strand, the only way that we can add new bases on is by adding on to this 3' end. We can't add it on here. So as the DNA opens up, we're going to have to work in sections. So we're going to make this part, and then we're going to see we're going to have to jump backwards and then open up this and make this. And then we're going to jump back and work backwards, and then eventually we'll jump back. These are going to be called Okazaki fragments, and I'll show you on the next 2 slides a little bit more about them. So elongation, this is where the big workhorse DNA pol 3 is going to do this. So our primers are laid down in green. Now helicase is going to keep opening up more bases, all right? So just follow him. He's going to keep going, following that direction of replication. Now this purple jelly bean right here, this is DNA pol 3. He looks for those RNA primers and then he hops on because that's like his, hey, that's where I go, and he hops on right there. And what he's going to do, he's going to go in the direction of replication that he needs to add on to the 3'. So on this top one, which is your leading strand, he's just going to keep going. So if you look at this bottom right here, so this is part 1, part 2. So we're extending. He's going to keep going and adding and adding. So your top strand right here is your leading strand. Now look at what's happening on the bottom. So he found his primers, and he needs to add them on new ones to the 3' primes. So he's going to go this direction on the bottom. So he's going this way. So he's going to keep going and going. Now he's going to keep going until he gets to another stop point. But look what's happening right here. So as helicase is opening up more and more DNA, this is the new stuff that we have to go back and copy. So let's see how we do that. So what's going to happen is DNA pole 3 is going to go and get to our stopping point, and then he's going to jump off. Now, as each of these new fragments open up, RNA primase is going to lay down new primers. So let me go back. So see how this is open? It's not shown, but RNA primase will hop on right here and lay down some primers and leave. So you'll see a new segment of green. See that new segment right there? Now DNA polymerase, what it did was pretty much hopped off and went backwards, right? And then he's going to keep going till he gets to right there. And he's going to keep doing this over and over again. Copy this, then he went back and did this one, and he's going to keep going till he gets to right here, and then he's going to keep hopping. So RNA primase has to keep laying these primers down for him. He keeps looking for those. So it's like he's playing reverse leapfrog. So if we talk about the order that these were done, he made this one first. Oh, I need my pencil. So this one was done first. He made this one second. He did this one third, and then this next segment will be segment 4. Each of these are his Okazaki fragments, so he pretty much is working like this. He gets to the end, and he just keeps going. My keyboard got in the way there, sorry. Boom. And then he'll get that in, and it just keeps repeating over and over again. So helicase will keep going and going and going, and this leading strand will go, but the lagging strand has to do this little leapfrog game right here. So we're going to need to stop it. So this is where the last 2 big enzymes come in. So all the bases have been added, all right? DNA pole 3 is done. I'm not going to go into detail talking about telomerase and how it wraps up the ends. That's more detail than we need to get into, but I'm just going to focus on these 2 guys. So DNA pole 1, or delta, what his job is to do is he's going to find those RNA primers. So you remember those RNA primers, the little set in green? We need to get rid of them. So at DNA pole 3, or delta is shown in this figure right here, boom, right there. He's going to find the RNA primers, and he's going to go through and he's going to cut them out and replace them with DNA nucleotides. So if I go back here, anywhere where he sees green, he's going to go and cut those out and replace them with blue. So what he does is he goes, cut out what's in there. Now the problem that we're going to have is we're going to have to make that phosphodiester bond between one segment to the other. He can't make that bond. It's just something about it. So that's where the last enzyme, DNA ligase, right here, is going to go through and fix that little part right there. So he's going to go through and add in, create that phosphodiester bond to make it a continuous phosphate back. So I know it seemed really confusing. Watch these. Super, super helpful. If you have any questions, let me know. But that's DNA replication in a whole.